new beginning for freshwater snail identification, or codiad wyneb if you're a Welsh speaker. That was the title of the project that we created in order to get a grant from the National Lottery Heritage Fund here in the UK to be able to basically do the work and produce the book. So it's a kind of grant getting title, not so much a real statement that we really are making a new beginning. But what we have tried to do is bring everything up to date with British freshwater snail identification for Britain and Ireland, and produce a new identification guide. And just about managed it, nearly there. The acknowledgements I'll put in at the beginning. I'll mention people again. Lots of them are here today. All the people who are in green are members of the society. And so it really is true that this project would have been absolutely impossible without the Conchological Society of Great Britain and Ireland, either members, current or past members, who actually collectively contributed a great deal to what we know about these species. Harry Powell, we've heard about. He was the project officer, so most of the grant was used to pay him to come and work at the museum for 2 years. And the other partners, including Martin, who's the current president, Mike Dobson, who helped produce the key, Hannah Shaw, who's involved with freshwater mussel, pearl mussel conservation through the Freshwater Habitats Trust, and also Rebecca Farley-Brown at the Field Studies Council. They've all done a great deal of work on this, and Martin was not paid a penny, but he's really bust his gut on all sorts of different aspects of it, and his expertise was particularly special for us. Many other people have contributed, so most of the pictures I'm going to show were ones I've taken or Harry took or that are from the book. But a few of the pictures are from other people who've sent them in, quite often at quite short notice, so we're really grateful for that. I hope you like freshwater snails, because I'm going to go on about them for for a while. This is the book, and I'm not going to do like a kind of run through of the whole book, but I will probably cover most of the things that you would expect to find in it when it finally comes out, which will be soon. It's the best I can say. We had the first proofs back from Field Studies Council this week, and we're going through them to do corrections. So once we've done that, they can get on with printing it, and it'll be available for sale from the FSC website, along with many other excellent guides to different groups. To give a bit of a structure to the talk, I've gone for I've tried to divide it into history, comedy, and tragedy in the Shakespearean sense. So the comedy is not as funny as you might expect it's going to be, and the tragedy is not as bad either. And I won't end on a tragic note, don't worry. It's just a loose structure. So here are some of the history aspects. Freshwater snails. Many of you and many people all over the world have been collecting freshwater snails almost habitually since prehistory, if not before. They're irresistible things to pick up on the margins of freshwater environments, or you might collect them for more useful purposes. But this is a collection that we got thanks to the help of Martin in particular. Is the Ed Bishop's collection, which was worldwide in scope. It's one of the best collections I've ever seen, but it also included British freshwater material. And we've used this and other collections by members like Barry Colville, Brian Coles, and past members like H.E. Quick, and even before the society existed, John Gwyn Jeffreys. So we've drawn together over 50 collections in getting things that we could use in this identification guide. And also photographs as well. The photos on the right by the late Derek Rans, who some of you may have known well. And he took some fabulous portraits of these tiny snails underwater in low light conditions, which is difficult enough to do in the digital era, let alone in the era when he took them on 35mm slides. And that collection, which Peter handed over to the museum, we've had some of it digitized, and we've been able to use those images, which has been a real help. And perhaps most importantly of all, the distribution maps, which largely are drawn from the Conchological Society's own efforts. So this data is from the NBN Atlas in the UK with the help of Sophia Ratcliffe, and also the Conchological Society's data from the Republic of Ireland as well, which Evelyn kindly helped me bring up to date for the book. And we've divided that in the book into 3 different eras. So not only can you see the distributions, but you can get a sense of how things have changed. And we might not be quite up to butterfly atlas kind of standards of things, but it does show how powerful our data can be when it's been assembled over such a long period and by so many people. So on the left, you've got an example of a really widespread species. That's the wandering snail, which now is supposed to be called Amgueddfa baltica, but a lot of people still call it Limnaea peregra, which is an exact synonym, so it's close enough in the UK. That's a very widespread one. And you can see it occurs in almost all the squares. Mixas glutinosa, sad story. It's one that's declined enormously in Britain and in Ireland too, although it's doing better over there. It's only known from a single site, but hopefully the colours give you a sense of that on the maps. And the dark brown dots are all 21st century records. So they're after the Conchological Society produced the Atlas by Mike Kearney in 1999. So it's a whole new phase of records that we're showing on the maps. And the one on the right is an example of a species which appears to be spreading. It's been recorded from lots of new areas since 1999. And how it's spreading, we don't really know, but it certainly seems to be. So snail distributions change, and that's one of the things that we needed to update in a new guide. Also, the world literature on freshwater molluscs has bloomed. Lots of new guides around. Some of you might have the Polish book, which is pretty good. It's in English and it's quite useful quite widely. Peter Gloyer produced a new book in 2019 covering the whole of the Western Palearctic, which is very good, very detailed sort of monograph on all the species, but it's perhaps not ideal for British beginners because it includes many more species than we would need. What many people still use, I expect including you, some of you, is McCann's key, which dates back to the 1960s. And although it was updated in a couple of subsequent editions, it hasn't changed that much. And this key is actually really good. It's still useful, but there are quite a few omissions in there. It doesn't go into much detail, and there are additional species that we've got since then. And some of the names and distributions have changed quite a lot. So it does really need to be replaced. So we've kind of tried to supersede this. And a few years ago, there was an attempt to cover the identification aspect of that, which didn't quite get off the drawing board, but the late Pat Hill-Cottingham, who some of you know, produced one which used this very popular method of a dichotomous key laid out like a kind of flow chart for identification. So we, as part of this lottery funded project, we wanted to involve ordinary people and beginners in particular in developing the key so that we would get something that was really going to be useful and accessible and easy to start with, but that would also work for the whole fauna and for people who had a bit more experience. So we had a series of workshops around Wales. They were all in Wales, but we had a few people come over the border, I'm sure, for them. Where people could have a go at different keys. And Harry, who you can see in the pictures here, took really careful note of like which of the keys were the most popular and what aspects about them people particularly liked. So with Mike Dobson's draft key, we kind of converted it into what was by far the most popular format, which was the flowchart one, a bit like Pat Hill-Cottingham had used. And that's been laid out for us by the Field Studies Council. So it'll appear in print, so that's why there's a dotted line down the middle, and it will work like that. There's lots of different sections of color coded, but it just asks you yes/no questions. Occasionally you have a species pair which we couldn't decide on a satisfactory way to key apart using a dichotomous key. So like those two Ficellas on the right there, they appear in the same box because a key isn't really going to cut the mustard there. You have to look at the species accounts. And of course, we say in the book, it's quite legitimate just to look through the pictures and then go back and check certain things using the key. That's perfectly okay. We did a lot of other public events as well. It was quite a schedule. This is me in Swansea Science Festival and Harry at the After Dark event that we did at the museum in Cardiff, where we were showing people freshwater snails from the collection and talking about different aspects of the project. Always good fun. We also did a little exhibition. It's very small. It's only, only covers one small wall, but even a small exhibition takes a bit of work to put it together. And on the right-hand side there, you can see the Conchological logo and all the other partner logos are on display in the museum, which does actually have, when it's open, quite a footfall. So many people will see it and have seen it. We made these models of some of the species. Since they're so small, it's good to kind of enlarge them so that people can see them and what the living animals are like in real life. And then at the bottom there, we tried to make a little diorama. We squeezed it in about the kind of things you might find in the bottom of a river. We also involved volunteers throughout the project. All the Conchological Society people who contributed count as volunteers, but we also had on-site volunteers. So there's Harry with Mike Tynan and Helena Neftayova. And Mike was a real stalwart. He helped us curate large parts of the collection, looked up all sorts of old localities and things. And he also came out on some field work with us, not just at Cardiff Castle, but a bit further afield as well. So, I mean, he was a diamond. This was an event or a series of sessions that we did at another museum site, which we called Snail Safari. And we had local school children who were on particular schemes because they were at risk of exclusion from the school system. And we did some pond dipping and snail identification with them. And they seemed to really enjoy it. A lot of them had never done pond dipping before. So it was their first experience of it. And they particularly liked doing things like messing about with the dredge and hauling out tadpoles and sticklebacks and things. So it's always a joy. We developed a game. This was largely kind of Harry's ingenuity, really, that we called Guess Whirl. I guess people know the kids' game Guess Who. It's like an elimination thing, and you get a mystery person. You have to describe their face, like, with yes/no questions. Have they got a beard? Do they wear glasses? To eliminate people in groups or one at a time. And we realised this would work really well with taxonomy and identification. So we did it for all the snails. And it actually worked really well. We turned it into a card game rather than a flip one. But we thought that was quite successful. And you could download these cards from the museum's website, although you have to print them out yourself. And you can play it. And that's my daughter on the right when she was 8, so it works with 8-year-olds. And the school kids seem to enjoy it. Also part of the public engagement aspect, we had to produce tweets and send out tweets all the time there on the left. But it was nice that we had some payback from engagement as well. And this was something people sent us in unsolicited. It was an illustration that somebody produced, an artist called Melanie Gale, after a visit to the museum. And she saw the model snails, and her imagination led to this design about the snail going on an adventure after dark in the museum. So very amusing. That's Valvarta pissinalis, as you can tell from the operculum and the weird snout that it's got. So by comedy, the comedy section, I kind of mean it in the Shakespearean sense. So it's mostly about mistaken identity rather than being actively hilarious. Fieldwork for freshwater snails is always a pleasure, really. You never know quite what you're going to find. So you can have a ditch like the one on the left, in which you could find 10, maybe even 12 species, a really diverse fauna. Or you can have a lovely verdant pool like the one on the right. It's called Pysgodlyn Mawr, that place. It's near me in South Wales, which means big fish lake. And you might expect it to be incredibly biodiverse, but it's not as good as it looks. And I only found 2 species in there. So this element really makes it worth looking in new places, because you never know what you're going to come across. So we did as much field work as we could over the project time span. It was only 2 years, so we had to squeeze it in where we could. We're in North Wales there on the Dee estuary, looking for intertidal snails on the salt marshes. We spent quite a lot of time on the project on these ones that live in kind of marginal environments, which are between the sea, fresh water, and the land, where several of the species are found. This is an extreme environment, the sea cave in Pembrokeshire. We were wearing cycle helmets in there, of course, to protect our heads, but there's one particular species, Paludinella, which is almost marine, is almost terrestrial, and almost freshwater, but this is one of the places where you can find it. Just by way of an example of some other places we went, this is a set of canals in Wales. We looked in every canal in Wales and in other places as well. Canals are quite interesting because they've got a strong historical background, and much of lowland England and lowland Ireland is connected together by a canal basin, which probably has a lot to do with the distribution of certain species. And here in the Glamorganshire Canal near Cardiff, June Chatfield found viviparous, the river snail, back in the 1980s. And we've had a really good look there, but it seems to have disappeared entirely, possibly concurrent with the gradual overgrowth of this canal, which is no longer used. I thought I might have found it briefly at one point when I found those snails on the right, but they are Valvarta pissinalis and not baby viviparous. So I've learned a lot through this project, and you know, I didn't start off by knowing everything, that's for sure. Swansea, sorry, there's a Tennant Canal in Swansea, and the Neath Canal just up the road. We had a good look there. Again, large parts of this are completely overgrown and taken over by birds like this very fierce goose that, that did its best to chase us away. Animals were one of the biggest hazards we encountered. Only one person fell in the water on one occasion, which was me, and no harm was done, so it wasn't too bad. The object that Harry was throwing into the canal there was the dredge on the left. So this was a homemade dredge. I made it with bits and pieces you could buy from Wicks, and it's probably, not including the cost of the bin that it's made out of, it's probably less than a tenner. But we used that quite successfully. It's a bit improvised, but it's light enough to be able to throw, and the mesh is a good size to be able to catch larger snails at least, and mussels from soft sediments. So it worked reasonably well, and we certainly used it a lot. But the absolute necessity, if you're going to look for freshwater snails, I think, is the sieve on a stick, which is a kitchen sieve, that thing in the middle, attached to a pole or broom handle with a couple of Jubilee clips, like hose clips. And those things are really useful, I think, often more useful than the more expensive FBA type nets you can get, although the mesh size of the sieve is, of course, larger than the sieve, so you miss some of the tiniest things. But a very useful thing to make and to have in the car or by the door. This is the true Swansea Canal. There's only a tiny segment of it left, and it's very overgrown in this part. But it's absolutely infested with these snails, Potamopergus antipodarum, which, as most of you probably know, is actually from New Zealand, but is almost ubiquitous in Britain and Ireland. It's one of the most widespread species of all, especially where there's a bit of flowing water through the river. And here in the Aberdare Canal, another place, haven for wildlife, but snail-wise is largely dominated by these Potamopergus. And there's a sunken log here, and there were so many on there, they were like flies on a corpse. They're just absolutely abundant. This is what happens when that process of overgrowth of a canal goes really far. This is Newport in South Wales. And as you can see on the right, some of these canals dry out altogether every summer because, I don't know, the bed is cracked or something, or the amount of vegetation just sucks all the water out. That actually suits some of our snails, which need periodic drying to thrive or to dominate, but it doesn't suit others. And there's a variety of weird plants in there, like this water soldier in the middle, which seems to be quite popular with these Planobarius, these great ram's horns, really like living among the fronds of that. But on the left, you can see we've got parrot's feather, Myriophyllum, which is one of these choking invasives that if you've got it in your freshwater water body, it can soon take over, and it can really reduce biodiversity overall. Still going through the canals. We haven't found any viviparous by this point, so we kept looking. This is Harry planking in the Brecon Beacons and the Brecon and Monmouthshire Canal at Talabont. It's got this kind of chocolate river effect, which is typical of a working canal in the UK. The bed is stony, it's got very little plant growth in it, and it's really turbid because basically the mud is stirred up by the boat traffic. And we didn't find very much in there, apart from these Ficella gyrinas, which are kind of they're about an inch long. They're quite big snails. And like the other physids, they're very active. They're always on the move, and they're making mucus strings, or they're climbing up surfaces. They're really quite lively animals, so they were quite popular when we put them on display in the museum as well. This is the Llangollen Canal. It's a World Heritage Site in North Wales because of this famous aqueduct where the canal goes over the River Dee. So I was desperate to have a look in there to see if there are any snails. And my wife and I used that same dredge, and we pulled it all along the aqueduct, and we didn't get anything because it was a working canal. But if you find these little backwaters, like the one on the right, and I think I'm right in saying this canal is connected to the English Canal Basin. So, you know, anything could occur in there. But in these little backwaters, you get a bit more of a buildup of plants and snails like these. This is a white ram's horn and another American Frisia species. Then finally, we found what we were looking for in the Montgomery Canal in Welshpool on the border with England. And we're right outside the Powisland Museum there. It's like Disneyland, but it's all about Powys. And we found these muddy trails on the bottom. And you can see at the end of that muddy trail there is a viviparous snail, and there's a live one at the bottom. So we finally found them in Wales. So they're not completely extinct, but they do seem to have disappeared from a lot of their former habitats. We kept these in captivity in the lab. So we had little tanks running for almost the whole of the project. I think Harry's still got some of them now, actually. And we observed pretty much the whole life cycle of viviparous. You can see them mating on the right, so they have separate sexes, and the males have this stumpy right tentacle, which they use to fertilize the female. And then, as their name viviparous suggests, they give birth to live babies rather than laying eggs. Another interesting thing about viviparous snails, and also Bithynia and some other freshwater snails as well, is that they don't have to graze using their radula like most slugs and snails do. They can just lie on their back, like that one in the kind of top left. They lie on their back, open up the shell, and they can filter feed by opening up the siphon on the right-hand side of their body. So they've got different ways of being able to extract nutrients from the water. Interesting animals. That's one of the biggest ones. At the other end of the size scale, you have little tiny things like these so-called spire snails, which, a bit like Ptamopergus, can occur in vast numbers, but generally on the marine side of things. And some of them are so strictly marine, they're almost marine organisms. And we've had a big debate in the COGSOC about whether they should be on the marine list or the non-marine list, or which of the two recorders should handle the records. And sometimes it's clear, sometimes it's not so easy to decide. Most of these we could find ourselves, but I did have to get live Hydrobia acuta neglecta. We had to ask Simon Taylor to send us some, and I thought he would send them from his local sluice in Essex, where he's recorded the species many times, but he actually sent them from the Outer Hebrides, which was very good of him. And we DNA barcoded all the species in the study, and generally speaking, they matched exactly what they were meant to match. So we're pretty sure that the specimens we're showing in the book are authentically identified. This is a much more obscure one that Martin has tracked down to just a single site in the UK. It's in southern England at Thorney Island, quite near a military base. It's really only very subtly different from the other spire snails, but if you see it in real life, you immediately realize, oh, this is distinct. There's no doubt that it's a different species. And it's very difficult to express that kind of thing in written form, whether it's online or in a book. You can give photographs, but it's difficult to capture that essential difference, let alone turn it into an easy key for beginners. So we've said as much as we can about these species to try and make that easier. But we DNA barcoded these, and yeah, lo and behold, they are semi-salsa. Apologies if I said Heliobia stagnorum, it's semi-salsa stagnorum. They are what they're supposed to be. This lagoon is a very weakly brackish lake, which is not connected to the sea, but it's very close to it. And it was a fun day where Harry and Martin and I used this inflatable boat to paddle right out into the into the reeds there to check that it occurred right throughout there. And the airbase didn't seem to scramble anything, so I think we got away with it. This is the inside of a hothouse. I think it's Oxford, and the photo is from Tom Walker, who's done a really thorough investigation of the major hothouses in Britain, a large number of which have got these ponds. And even in like the kind of 20 years I've been involved with snails, things that were initially thought of as greenhouse species have spread out into the wild, into gardens, and they're becoming more and more common. So it may only be a matter of time before some of our hothouse freshwater species, or more of them, do the same thing. Here's a couple of planorbids. They've certainly done that in southern Europe and in warmer places. The Melanoides tuberculata, the thing on the top left, is a tropical percolate snail, but that's already been released into the outdoors in Britain, into the cold waters of the River Tyne, of all places, where Adrian Norris recorded it a few years ago, and it survives in water that's heated by industry. So it seems almost anything is possible. On the right, we got some succinate snails, some amber snails there. And anyone who's ever been looking for freshwater snails in Britain has probably come across amber snails. And in fact, one of the first parts of McCann's freshwater snail key asks you to look at the eyes of the snail and see whether the eye is on the end of the tentacle or at the base, which is all very well if you've got the live animal, but if you don't, then people get thrown and they don't know what to do. So we've included a bit of advice on how to separate amber snails from true freshwater ones. But one further fly in the ointment is in continental Europe, this American species, Pseudosuccinea columella, is becoming more and more common. Again, it's starting in greenhouses and aquaria, and it's spreading out from those environments into mild places. And irritatingly, its shell looks very much like a succineid. So we put this in the book, but just with a rider, a warning on it, saying that it hasn't yet been recorded from Britain or Ireland, but maybe one day. So when we did have cases of mistaken or confusing identity, we wanted to at least do some DNA work to be able to be a bit surer about what they actually were. So if you've had a coronavirus test, which involves PCR, then it will have gone through a machine a bit like those ones on the bottom left, probably a newer and larger one than those, but those are PCR machines. So you do a PCR reaction to amplify the DNA of interest, and then if it's worked, you get a bright band on your gel under ultraviolet light, like those on that square gel there. And if it hasn't worked, you don't get a band. And it can take a lot of trial and error and optimization before you can be sure that you're going to get good results, really. So you can see on that gel that most of them have worked, but a few of them haven't. And I think some of the ones that haven't on there were the Corollas achronicus that we tried from older collections. And looking for differences in the sequences is something that's best done with the aid of computers. So you line up the sequences and you can look for differences in the DNA sequence as highlighted there. So here are a few of those cases. I see Ron Carr is at the meeting as well, and so is Ian Killeen, and they helped British workers separate Stagnicula fuscus from Stagnicula palustris, these marsh pond snails that to be divided into several species across continental Europe. There's loads in Peter Gloyer's book. And Katrin Schneebs and others have done DNA work to back this up. There are definitely two anatomical forms in the UK, so you can't really tell the shells apart. There doesn't seem to be any reliable way to be able to distinguish them from their shells. But if you cut them open and you look at the penis and the prostate, then there are definitely two different forms. And the fuscus one is more common, and it seems to be the only one that you regularly find in the north and the west, whereas the palustris one is, it's the only one you regularly find. The palustris one is mainly found in the southeast and in kind of more diverse habitats. But when we looked at the DNA, we didn't get two different groups. All the British and Irish sequences came into this grey clade in this this genetic tree here. And I think perhaps the explanation for that is that rather than having two completely distinct species, we've got Fuscus, and then the other ones are Fuscus palustris hybrids. Now that hypothesis comes from Schneeb's et al. Paper, where they recognize palustris as a completely distinct species. So there it is at the top there, but one that could nonetheless hybridize with Fuscus in some places. So they had other sequences, some of which are in this tree, in that grey clade. So it's a really confusing situation. It doesn't really make things easier for beginners at all, but we've tried to explain that as best we can in the book, and we've still kept these ones as two separate species since they are distinguishable from their anatomy, and they seem to have different distributions. You can complain about these things at the end if you disagree with my interpretation. This is Myosotella, these mouse-eared snails that live on those salt marshes and other almost intertidal habitats, often in quite low salinity though, so they're almost freshwater animals. And there's a long-standing debate as to whether we've got one species or two species around Britain and Ireland. There's denticulata and there's Myosotis, and the denticulata one is usually got a thicker, paler shell with many more teeth in the aperture, and Myosotis is brown. It's got less teeth and it's often a bit bigger. But if you ask me, I think there's a continuum between the two forms. We looked at lots of collections, loads of specimens, and other people who've revised this group, like Tony de Frias Martins, have also concluded that the two species aren't actually completely distinct. People sent us specimens. Roy Anderson sent us some from two different habitats in Northern Ireland and of two quite different looking forms. Ian Smith did a great job. He's found denticulata type 1s on the D estuary. He sent us some of those. And Bass Payne sent us some from the Scilly Islands as well, where some of the habitat differences that are traditionally associated with these forms seem to be, if not reversed, then at least broken down. And I sequenced about a dozen of those, and I just found they all fell into one clade with not very much variation within it. It's less than 5% differences between the sequences. So it doesn't really strongly suggest that there are two species. I think there's probably just the one. And so that's how we've treated it in the book. But again, I've tried to condense all this down into the text and the species account so that people can study it further if they want to. Another nuisance species pair that people have been interested in and arguing about for a long time is Anisus leucostomer, button ramshorn or white-lipped ramshorn, which is one of those species that really likes temporary pools. There's a pool there which dries out. I went there this morning actually. It's got water in it at this time of year, but it dries out every summer, and it's got lots of Anisus leucostomer in it. In continental Europe, there is definitely a separate species called Anisus spirorbis, but for well over a century, there's been a debate about whether spirorbis also occurs in Britain. And I have to say that I don't really think it does. You get forms like these ones on the top right. That's one Peter Topley collected for us before this project began, actually, in about 2015, from Wilden in Bedfordshire. And that's about as different as they ever look to a more typical one on the left, which is from Stockholm Island in Pembrokeshire, which had previously been recorded as spirorbis by no less than a great naturalist like Charles Oldham, who collected them from there before. So that's why I went there to try and get some more and compare them to the ones from Bedfordshire. And the DNA of those two forms is actually very similar. Again, it's less than 5% different, so not really suggestive of two separate species. Peter Gloyer in his publications has provided extra evidence in favour of there being different species on small differences in the shape of the shell and the width of the last whorl compared to the width of the penultimate whorl. But if you put all this material onto a single graph, it looks to me as though there might just be small and large forms of a single species closely related to size, that shape difference. There's an anatomical difference. Again, it comes back to the prostate, the number of lobes you can count in it. And the British ones have got an intermediate number of lobes between those found in Leucostomer and Spirorbis, or they're clearly Leucostomer and they've got loads of lobes. So again, I'm not really convinced that there are two different ones. Of course, anything's possible, and somebody might find some really, you know, outstanding examples of Spirorbis somewhere, which turn out to be different, because we keep introducing new species from the continent anyway. Corollas levis, even this one's not as simple as it looks. And this is a species that's always at the end of the key. So it's a good example of a species which doesn't have any distinguishing features of its own. It just doesn't have any of the clear ones that the others do. So it's difficult to describe what makes it unique. It's called a smooth ramshorn, but it's not really completely smooth. It tends to have a round aperture. It's small. It's got loosely coiled, quite rounded whorls. And it's got a strange distribution. It seems to occur almost everywhere, and yet isn't concentrated anywhere, and is always rare. It's very local and sort of almost over dispersed. And it likes different types of habitats. It doesn't like rivers or flowing water very much, but you can find it in any sort of pond or lake, it appears. We found it in newly created ponds as well as really mature ones. On the continent, continental authorities at this talk can correct me if I'm wrong, but they recognize two species. They also recognize something called Corollas parvus, which was described for the United States, and that is considered a spreading alien or invasive species that you can tell apart by a minute difference in the anatomy, or by this thing indicated by the white circle there on the left, which is that the last whorl sticks up a little bit. Sorry, the penultimate whorl sticks up a little bit over the next whorl. I'm not sure if that's a really massive difference, but the species have always been kept separate. But we sequenced specimens that Roy sent us from Northern Ireland. Adrian Sumner provided some from 2 places in Scotland, and then we found it a few times in Wales. Sadly, we couldn't get any from the type locality for Corollas levis, which is in Northumberland, but we may be able to in future. Anyway, all these sequences came up as a really close match to North American ones. So it's difficult to know exactly what's going on here. Either Corollas parvus in North America is very closely related to European Corollas levis, or the British fauna has been secretly invaded by American ones, and we just haven't truly got to grips with it yet. That's perfectly possible. It's happened before, like with the Frisia limpets, covered large parts of the globe before people realized they were invasive. But there's no doubt that Corollas levis is native to the UK, because it's in the subfossil record as well. So mysterious and surprising. On to the tragedy. So one tragedy that continuously affects freshwater snails is that they play host to these parasites, which we only really pay attention to because of the effects they cause on livestock, or in some cases people, but in this country mainly livestock. Now I learned about the sheep liver fluke life cycle. I think Imogen did as well. I remember us talking about it once. Every Welsh school child has to be taught about the sheep liver fluke at school. And I just kind of assumed that it was a problem of the 1950s or something, and it was like a fairy story they told us to make sure we understood how far technology had moved on. But they're very much still with us and with farmers, these liver flukes. And we've got extra flukes as well. We've got rumen fluke and other ones. So this is people at Aberystwyth University, Rhys Jones and Chelsea Davis, and they're studying, even today, ways to detect and control liver fluke on Welsh farms, where it causes serious economic damage and affects the welfare of livestock quite considerably. I've got a very particular gripe with liver fluke, in that it really spoils the anatomy of the snails I dissect. So this is there's a healthy radix auricularia there on the left, and I cut that open so I could look at the penis and the bursa and make sure it was the right species. But on the right there is one that's heavily infested with fluke cercariae. That one's from near Belfast, actually, but almost anywhere where you collect large limnaeids from, you get a lot of them that are really absolutely taken over by these parasites. And they shed the larvae of these parasites into the water column at an amazing rate. There's a huge biomass, actually, and there's almost a whole ecosystem built on invertebrates that feed upon the emerging cercariae larvae of the flukes from the snails. So it's a very underappreciated part of wildlife in the UK. There's a bit of a tragic story about this snail, I fear. It's Marstoniopsis insubreca. The common name, I can't remember it. I'm not even sure if it has one. It's a very tiny little snail. Found mainly in canals and a few rivers in Britain, but there are places like the northwest of England where it hasn't been seen since the 1970s. And it was last seen in East Anglia in about 2008. And nobody's seen it since. So the photos of the live animal there I got from Jonas Roth in Sweden, where the species is still common, but it's possible that it has completely died out in Britain. Hopefully, with a bit of extra information like we're providing in our book, then it might stimulate people's attempts to look for it. And then you get a good news story if somebody rediscovers it. So here's hoping it will turn out that way. A real tragedy, I've learned a lot by doing this project. You can go to lots of places, particularly all these fishing lakes, which are lovely places to spend an afternoon on the bank, but they're terrible for pond dipping. There's very few invertebrates in them at all, and in the turbid, kind of over-nutrified water with too many fish in. This is another lake where I just found 2 species, both of them American and invasive. So there's the Frisia that I mentioned before, the American lake limpet, now called Frisia californica. It was called Frisia waterii when it was believed to be South African at one point, but it appears they're all California originally. And Ficella acuta, the thing on the right. Ficella acuta, it occurs in almost any garden pond or fish tank, and it's becoming more and more common. Ian and Evelyn found some in the southwest of Ireland recently, and that was, I think it's all right to say so, I think I was in the Killarney National Park. So, you know, none of our environments are really safe from this American phycid. And there's some really interesting suggestions from France that the spread of alien phycids can have an effect on native species, in particular the mud snail, Omphiscola glabra, which is quite rare. It's declining throughout Europe. But they found that the phycids were associated with a decline in the numbers of Omphiscola in ditches and ponds. So they don't really know why, but it's possible that these American things are outcompeting native biodiversity. This is a whopper that Evan Jones came up with. That's Evan's hand there, I think. He found it in the Pevensey Devils National Nature Reserve. So somebody had introduced this Chinese monster snail, Chinese mystery snail, into that ditch there. And Martin and Evan are now working with the authorities to try and eradicate this species. It could conceivably cause problems, and the only opportunity to get rid of it is now before it's spread any further. It looks a bit like a viviparous, but it's bigger, it's greener, and the juveniles look different. If you see it, it's actually quite unmistakable. So all these invasives and many others have added a lot of weight to the Check Clean Dry program in the UK, which has been quite successful. If you go to some freshwater habitats, particularly those popular with tourists and boaters and anglers, you might see these Check Clean Dry signs, which are to encourage people to clean all their equipment, like our dredge and our nets, and also people's wellies, to make sure you don't spread either these species or their pathogens and parasites between water bodies. I think perhaps, you know, given the last couple of years, I think people may take sort of this kind of environmental health hygiene restrictions a bit more seriously. Maybe the saddest story of all is about Corollas achronicus, the Thames ramshorn. This is a native species with a subfossil record. It's the only British species that's confined to a single river catchment, which is the River Thames. And there's lots of accounts of it being very common in the River Thames in the early 20th century. It's quite distinctive. It has this fringe of hairs around the edge. And although I couldn't get any DNA from any British specimens, I think it probably is conspecific with the European ones, and it is a distinct species. It's not the same as Corollas albus or anything. But the last person to see it alive who is here this afternoon, that's Ian Killeen. And I think he last saw it in about 2010, so it hasn't been seen for a decade alive. Tom went out to look for it, as did Harry and I. We went to several places. We couldn't find it. Tom couldn't find it either. All you get is the occasional dead, empty shell, even at places where it was common a few years ago. But what you do find are these guys on the right, the American signal crayfish. They may not be directly responsible, but we've noticed that places where you find a lot of crayfish, you tend to find very few snails. And we didn't actually encounter signal crayfish anywhere else in the project except at the Achronicus sites. And they were absolutely abundant there. You couldn't help catch them. They do eat snails. That one on the top has just finished eating a snail. A much more systemic problem. I'm not a freshwater ecologist, but I do understand that if you have too much nutrient in the water, then you get algal blooms, and that kills things off. It's bad for all wildlife, but it includes snails. Eutrophication, and that's the Chichester Canal that Martin saw this eutrophication in, I think it was last year. And there's some evidence from the Conchological Society's data that not only have freshwater snails and bivalves declined more than their terrestrial counterparts. Charlie Outhwaite and others at the Centre for Ecology and Hydrology did a big study on various invertebrates using Society recording scheme data a couple of years ago. And they were using fancy methods to model species occupancy, but they found that the number of squares occupied by freshwater molluscs had declined considerably. And unlike other freshwater invertebrates, like mayflies and other insects, they hadn't recovered so well after waters were generally cleaned up in the 1990s, possibly because they're not very good at dispersing. So all very sad story, really. And after writing all this up for the book, which we finished in about November time, it was pretty miserable. This was the Christmas card I prepared to send to people, but my wife said that's just not Christmassy at all. I got a bit too obsessed with the snails and their and the threats to them. So I tried to Christmassy up with a bit of decorations, but that didn't seem to help either. So thankfully, winter is over. I've nearly finished the talk, and summer is coming in. Excellent. New life springs. This morning, I found these eggs in my pond. These are the eggs of Amgueddfa baltica, and we hope that our book is going to let people identify snails even before they hatch, because we've included a guide to eggs and spawn, also some of the eggs and spawn that you might find of creatures that aren't snails. But some of these are fascinating things in their own right. So in the middle, you've got the eggs of Bithynia, which once you've seen them are impossible to forget. They're like, it's like frog spawn, but it's a bit more like bubble wrap that's stuck to a leaf, and it's very tough, flexible stuff. And you get loads of snails, and they're just beginning to hatch out as we go into spring and early summer. The eggs of Anculus, a freshwater limpet, really distinctive. You'll find them almost everywhere you find the limpets themselves. And my particular favorite are the eggs on the bottom right, which are from Valvarta, the valve snails. They're like this amazing little snow globe full of green peas, and they produce these eggs really quite fast. They can lay one of these snow globes every day. Very, very small, but once you get your eye in, you start to notice them everywhere. And we have also included a bit of special help to get people to identify juvenile snails. We've tried to include as many photographs of authenticated juvenile shells as we can, but we've done a particular thing on these limnaeas where baby Omphuscola mud snails look quite similar to adult fluke snails, Galba. And last year's spring lockdown was actually a good opportunity to find the eggs of these snails and then breed them or rear them in my kitchen and take photos of the shells at different stages, so you could show the whole growth series. That's Hannah Shaw on the right, showing me how to look for mud snails properly, which I'm pretty sure I've been doing, but it really is quite a rare species that you can't find that widely. And Hannah and I don't really want to call it just mud snail, as many organisations do. We think it should be called a slender mud snail, because the word mud snail has been used for so many different British species over time. It's a bit confusing, so I should have said slender mud snail. So that's about all. I hope I've given you a sense of what's in the book and convinced you that it might be a useful thing, whether you're going out to exploring these freshwater paradises on your own, or hopefully as soon as possible in groups. I hope people are going to find it useful. But in particular, the message I wanted to get across is to thank the Conchological Society for helping, well, for everybody keeping their eyes peeled, and also for helping us keep our chin up over the last couple of years. And it's been a really good project, and I hope it's going to lead to better things in the future too. Thank you very much.
Right.
I've stopped sharing my screen, but I can bring it back if you like. You're all still there, that's good. There's a message, there's a question in the chat. Can I answer that?
Can't hear Martin.
That's from Rosemary Winnell to everyone. What were the eggs that you showed on 2 of the snails? I can't remember the name, but it was the one that Martin has found recently, a single site. Oh, yeah, that was the Semisalsa stagnorum picture. You quite often find these tiny little brown capsules on very small snails. I don't know what eggs they are. They are not the eggs of Potamopyrgus antipodarum, which is another snail species that lives in that lake, because that doesn't usually lay eggs. It's a viviparous one. We don't know. They could be insect eggs. Maybe somebody here knows the answer, but I'm not sure that they're snail eggs. You find them plastered all over things.
We don't think that those are the Semisalsa eggs then, Ben?
They could be, but you find very similar ones on other places as well where you don't get Semisalsa. So.
Right, and he keeps them in captivity. Right, other questions? I've got Ron with his hand up, first of all. Can I ask you to give your question, Ron?
Can you hear me?
Yep.
Yeah.
Yeah, excellent talk, Ben, and congratulations on the book.
Oh, thanks.
As you told me some time ago, it was a bit disturbing to hear about the DNA results for the Stagnocollas. And I think your explanation is probably quite valid about the hybridization. The interesting question is, which one came here first? Molluscs has got the southeastern distribution, which would tend to suggest that that's likely to be the newcomer. On the other hand, Fuscus seems more tolerant of pollution and has got, whereas Molluscs seems to prefer more refined environments and with densely vegetated, with a good range of plant species. So I don't know if you've had any more thoughts about it since we spoke.
Well, I agree.
I mean, the other thing was that there was another paper, which I'm sure you've probably seen, and probably predates the one that you mentioned, European paper by Bargs. There's about a dozen authors on it, which also separated European material into different species.
Yes, yeah, I know the one you mean. I agree with you. I think that does seem to be a real difference in their distribution and ecology. I don't know which one arrived in Britain after the ice first. Probably Fuscus, because it's spread to Ireland, whereas the other one hasn't. But
absolutely, yeah,
yeah, it's a really complicated thing, and it's a bit like with the slugs situation. In a way, we're lucky that we live on an island because we've got a small fauna which is a subset of the European one. But whenever something new is noticed or arrives, it's difficult to know like how much of the European diversity we've really got that match things up. So I've just followed the conclusions of Schneebs et al. About the species hybridizing. I think they're probably right. Yeah. And I'm just thankful that we don't live in like the northern Italy or something where there's many more species to worry about.
Yeah, yeah. I mean, I suppose it'd be interesting to carry out some DNA studies on material just across the channel in France and Belgium, just see how that compares with British.
Yeah, it could be. Yeah.
Anyway, thanks.
Another question, Tom.
Can I ask?
Tom?
Yeah, it's just on Corollas the Chronicus. You commented that it was last seen by Ian Killeen 10 years ago, but in my searches on the River Pang last year, I certainly found one fresh dead. Still had the periostracum on, it was still translucent as opposed to the long dead ones, which are opaque white. So I think there is still hope, and I'm hoping you're coming to join me sometime this year to have another look. But, you know, there is still hope.
That's good. So you think it was alive recently then, that one?
Well, I would have thought within the year, certainly. I'm not quite sure how long the periostracum goes, but it was still translucent.
All right.
You know, there's a chance.
There is a chance, yeah. I think that whole situation might be a good application for eDNA as well, if that technology works in a river. Unfortunately, I couldn't get any DNA from the live collected specimens that were quite old, but yeah, that might be a good way of finding out where it is. But Ian did these surveys for the Environment Agency in the past, and I don't think they've recommissioned anyone to do something similar recently.
I mean, this was looking in the areas that Ian did, but then I did find crayfish as well.
Yeah, they're easy to find.
Yep. Anyway, brilliant talk. Thank you. Much looking forward to seeing the book.
Thanks.
Okay, do we have anybody else wishing to ask a question? I'm trying to look at both screens and there's nothing showing there. Is that the full extent? Your last chance to ask a question.
Well, you can always contact me. I don't mind.
Right. Okay. Could you, if that is the end of that, could you all unmute yourselves, please? So I can see people muted still. Could you all unmute yourself? Right. Now, the reason for doing that, David, you need to unmute. David Hollyoak. I can't hear you. You're muted, David. He's talking, but I can't hear him. Right. Okay. Well, I'd like to thank you very much, Ben, for a fascinating talk. And so in the usual traditions of the Conchological Society, I'd like to offer you a round of applause, a virtual round of applause. We can all clap. Thank you very much. Deafening round of applause. Thank you very much. Right. It is now the time where we take a 10-minute break for whatever you'd wish to do.